Have you ever wondered why milk goes sour? Or how a tiny cut can get so sore? For thousands of years, people saw these things happen, but they didn't know why. They saw bread rise, grapes turn into wine, and they saw sickness spread through entire towns. They knew something was happening, something they couldn't see. They came up with all sorts of ideas, many involving magic or angry gods. But some thinkers, way back in history, had a different idea. They guessed that maybe, just maybe, there were tiny living things all around us, too small for our eyes to see. Consider this. Long before we had the cool gadgets of science, some very smart people were on the right track. In ancient India, followers of Jainism believed in tiny, unseen creatures living in the air, water, and earth. They were careful not to harm these invisible life forms. Later in the 1500s, an Italian scholar named Girolamo Fracastoro proposed that diseases could be spread by tiny seeds or spores. These seeds could travel from person to person, through the air, or by touching things. He didn't have a microscope to prove it, but his idea was a huge step forward. It was a thought experiment, a scientific guess based on observation. Science. Then, everything changed. In the 1600s, an English scientist named Robert Hooke built a microscope. He looked at a thin slice of cork and saw what he called cells. They looked like tiny rooms. He also looked at mold and described its structure. He was one of the very first people to see a microorganism. But then came Antony van Leeuwenhoek, a cloth merchant from the Netherlands. He wasn't a trained scientist, but he was amazing at grinding lenses. He built powerful single-lens microscopes that were way better than anything else at the time. He looked at everything: pond water, scrapings from his teeth, rain. And what he saw blew his mind. Leeuwenhoek saw a whole new world teeming with life. He called the little swimming, wiggling things he saw animalcules, which means little animals. We now know they were bacteria and protozoa. He wrote letters to the Royal Society in London, describing his incredible discoveries. But this led to a big question. Where did these tiny creatures come from? Many people believed in something called spontaneous generation. They thought life could just pop into existence from non-living stuff, like mice from dirty rags or maggots from rotting meat. It was a popular idea, but some scientists were skeptical. They wanted evidence. The debate over spontaneous generation was a big one. An Italian scientist named Francesco Redi decided to test it. He set up an experiment with jars of meat. Some jars were open, some were sealed, and some were covered with a fine cloth. Maggots only appeared in the open jars where flies could land and lay their eggs. No flies, no maggots. Later, Lazzaro Spallanzani boiled broth in flasks. He sealed some flasks and left others open. The broth in the sealed flasks stayed clear, while the open ones got cloudy with microbes. These experiments were powerful, but some people still weren't convinced. They argued that boiling the broth destroyed a life force in the air. Then came the one, the only, Louis Pasteur. This French chemist did a brilliant experiment that settled the debate once and for all. He used special swan neck flasks. He boiled broth in them, and the S-shaped neck let air in, but trapped dust and microbes in the bend. The broth stayed perfectly clear for months. But if he tipped the flask so the broth touched the trapped dust, it quickly became cloudy with microbial life. This proved that the microbes came from the air. They didn't just appear out of nowhere. Pasteur saved the wine and beer industry by developing pasteurization, a process of gentle heating to kill harmful microbes. Pasteur's work led to the germ theory of disease, the idea that microbes cause infectious diseases. Around the same time, a German doctor named Robert Koch provided the proof. He was a master at growing bacteria in the lab. He identified the specific bacterium that causes anthrax, a deadly disease in cattle. He developed a set of rules, now called Koch's postulates, to prove that a specific microbe causes a specific disease. Koch and his team also discovered the bacteria that cause tuberculosis and cholera. Their work was revolutionary. It changed medicine forever by showing doctors what they were really fighting. With the knowledge that germs cause disease, the next step was to fight them. A surgeon named Joseph Lister realized that many patients were dying from infections after surgery. Inspired by Pasteur's work, he started using a chemical called carbolic acid to clean surgical instruments and wounds. The results were amazing. The number of deaths from infection dropped dramatically. This was the beginning of antiseptic surgery. And in the lab, a Danish bacteriologist named Hans Christian Gram developed a new staining method. The Gram stain allowed scientists to divide bacteria into two big groups, gram-positive and gram-negative, which helped in identifying them. It's a technique we still use every single day. Just when scientists thought they were getting a handle on the microbial world, they found something even smaller. They discovered that some diseases were caused by agents that could pass through filters fine enough to trap all known bacteria. These mysterious agents were called viruses. It wasn't until the invention of the electron microscope in the 1930s that we could finally see these tiny infectious particles. This opened up a whole new field of study called virology. Understanding viruses became crucial for fighting diseases like polio, measles, and the flu. It was another reminder that the invisible world was more complex than we ever imagined. The first half of the 20th century also brought one of the most important discoveries in the history of medicine: antibiotics. In 1928, a scientist named Alexander Fleming noticed something odd. A mold, a type of fungus called penicillium, had contaminated one of his bacterial cultures. But around the mold, the bacteria were gone. The mold was producing something that killed the bacteria. He called this substance penicillin. It took a few more years for other scientists to figure out how to purify it and produce it in large quantities. But it became a wonder drug that saved millions of lives during World War II and beyond. Then came another game-changing discovery: the structure of DNA. In 1953, James Watson and Francis Crick, using data from Rosalind Franklin, figured out the double helix structure of deoxyribonucleic acid. This is the molecule that carries the genetic instructions for all living things, including microbes. Understanding DNA allowed us to understand how microbes work at the most fundamental level. It opened the door to genetic engineering, where we can change the DNA of microbes to make them do useful things, like produce insulin or clean up oil spills. Genetics gave microbiology a powerful new set of tools. As our tools got better, we kept finding new life in unexpected places. For a long time, we thought all life fit into two big categories. But in the 1970s, a scientist named Carl Woese was studying microbes that lived in extreme environments, like hot springs and deep-sea vents. By looking at their genetic material, he realized they were fundamentally different from bacteria. He proposed a whole new domain of life called archaea. These microbes might look like bacteria, but on a genetic level, they are as different from bacteria as we are. This discovery completely redrew the tree of life. So, where are we today? Well, microbiology is everywhere. It's at the heart of modern medicine. We use it to diagnose diseases, develop vaccines, and create new antibiotics to fight drug-resistant superbugs. We now understand that our own bodies are home to trillions of microbes, our microbiome, that help us digest food, train our immune system, and even affect our mood. Keeping these tiny partners happy is a whole new frontier in health and wellness. We've gone from thinking of all germs as bad to understanding that most of them are essential for our survival. Microbiology is also transforming agriculture and industry. Scientists use microbes to create healthier soils and protect crops from pests without using harsh chemicals. They engineer yeast and bacteria to act as tiny factories, producing everything from biofuels to biodegradable plastics to the enzymes in your laundry detergent. Microbes are used in food production to make cheese, yogurt, bread, and so many other things we enjoy. They are nature's ultimate chemists, and we are just beginning to learn how to work with them to build a more sustainable world. What an amazing thought! We are also using microbiology to take care of our planet. Microbes are the world's great recyclers. They break down dead organic matter and cycle essential nutrients like carbon and nitrogen through our ecosystems. We harness this power in wastewater treatment plants to clean our water. We use a process called bioremediation, where we use microbes to clean up pollution like oil spills in the ocean. Understanding how microbial communities respond to climate change is one of the most important areas of research today. They are tiny, but their collective impact on the planet's health is enormous. From ancient guesses about invisible life to the high-tech gene editing tools of today, the journey of microbiology has been incredible. It's a science that has revealed a hidden world that is not just all around us, but also inside us. It has saved countless lives, fed the world, and given us powerful tools to protect our environment. The story of microbiology shows us how scientific curiosity, careful observation, and brilliant experiments can change the world. And the best part? There are still so many discoveries waiting to be made. The microbial world is vast, and we have so much more to explore. Science rules.